Bismillahirrahmanirrahim. We're going to start the specimen paper 4, which I'm going to discuss here for the new syllabus 2023, which is going to be held in May, June this year. So I hope this is going to help you in understanding how to handle the paper 4s. And this, of course, used to be called paper 6, but now it is called paper 4. So there's not much change in it except for the numbering of the paper has changed. So this used to be in the previous called paper 6. Now it will be called paper 4. And the variant paper 4 too will probably be in Pakistan, but for different countries, it'll be different variants. So best of luck, and let's see how you all can understand this paper a little better and attempt it in this coming year. Now, before I start discussing the question paper, I want you to remember 4 points. Number one, read the whole question. You must read the whole question because if you're writing something in part A and then you're writing it again in part B, then there is something wrong. You are writing that you cannot be awarded a mark for the same answer. Number two, look at the marks for each part of the question. Are there 2 marks? Are there 3 marks? Are there 4 marks? How many marks are there? So you must think of the mark scheme points. If there are 2 marks, have you given 2 points? And then give another point because maybe you might not have worded one of the points correctly. So if there are 2 marks, give 3 points. If there are 4 marks, give 5 points. Then number three, always underline keywords. When you underline keywords, then you are emphasizing and your brain, your eyes and your fingers are working and you then focus on that point that this was the part of the question which I had to address. And number four, read the question twice. You must read the question twice because you then may have missed out something. Sometimes you miss out a certain part of the question. You're all now in an area, you are the generation which is makes a lot of careless mistakes. And a lot of careless mistakes cost you marks. And at times when I return tests which I have marked, and students say, oh, miss, I didn't see that. Oh, miss, I didn't read that. So you must read the question twice and read it very slowly so that you can understand it and comprehend it and then start writing your answer. As you can see, this paper 50904, this is a specimen paper and this is not the actual paper and this is for 1 hour and you are allowed all the questions. You can see what is all allowed is use a black blue pen, you may use a HB pencil, do not use an erasable pen or a correction fluid. That's very important that you know that. Do not write on any barcodes. You may use a calculator. The total marks is 40 and the time is 1 hour. So please remember the time is 1 hour, not 1 hour, 15 minutes or something like that. And suddenly you say, oh my God, I didn't know this was 1 hour. Now, starting with question one. Now, when I read the question, just look at the way I read it. Glucose is a reducing sugar. Benedict solution is used to test for the presence of a reducing sugar. Part A, describe how you would use Benedict solution to test for a reducing sugar in some fruit juice. Include a suitable safety precaution in your answer, and this is for 3 marks. Now I know students what they would write. Test with Benedict's, heat, add the fruit juice. And you see, it's very vague if you do the things like this. You must be very specific. I mean, I haven't written what the points I would have written, but I'm just giving you a guess of what you all would write. But you all must think of what would be the possible 3 marks. The 3 marks are that you took the fruit juice and add Benedict solution to the fruit juice. So add Benedict solution to the fruit juice. And somebody would say, Miss, well, that would be something that everybody would do. No, everybody won't write that. Because you might say just add Benedict solution. Well, I can't give you a mark because you didn't write fruit juice. So you see, you had to write the fruit juice, you had to write the Benedict solution. If you just say, okay, take the fruit juice and add the reagent to it, well, even then you don't get a mark. So I'm giving you all the possible wrong answers and also the correct answer. So add a Benedict solution to the fruit juice and then you heat. Now, if you were in A levels, I wouldn't have given you any marks for that because heat is not getting to any mark if you don't tell me the temperature. So it's 100 degrees centigrade, but you could have given me any temperature between 80 degrees Celsius to 100 degrees Celsius. I like you to put in the temperature because I think it's very important that you should know. Just heating is an O-level thing, but of course, I believe you should know a little more as well. And then the fact that they've said use a safety precaution. No, Benedict is a chemical, so you need to know that. Plus, you are heating, so the use of a water bath would get you another mark for the safety precaution. So use a water bath. Or you could have said something else, use an eye protection, or you can use goggles. Why are you going to use goggles or eye protection? Because if God forbid this spills on you, by mistake, you just, somebody's passing by and hits your elbow and it spreads into your face or something. So you use a chemical, for the chemical not to fall onto your eyes, you can use goggles or eye protection. But if you write something which is totally rubbish, then I'm sorry, you're not going to get a mark. So you realize 3 points. There were only 3 marking scheme points. You couldn't have given me anything else. There was no fourth or fifth point. You had to give me add Benedict solution to the fruit juice, heat in a water bath. If you said heat in a water bath, yes, you'd get 2 marks. It's not the sentences I'm looking at. It's the material which I'm looking at. If you said heat using a water bath, well, I would give you your 2 marks. 1 mark was for the Benedicts and the fruit juice. Second mark was for heating. Third mark was for a water bath. So if you said heat in a water bath in one sentence, you would get your 2 marks. Now, as you can see here, to detect the presence of reducing sugars in a sample using the Benedict's test. Now, you can look at this one. This is sort of blue. There's no reducing sugar in it. But as we keep on, as the reducing sugar increases, the color becomes green and then yellow and then orange and then red. Now, this indicates the quantity of reducing sugar. This indicates the quantity of reducing sugar. More the sugar, the more it will be towards this side. So if it is less, it's going to be just a trace is going to be green and then a little more and then more and then more. So this is a semi-quantitative test. It roughly tells you the quantity of reducing sugar. If I give you 2 samples, apple juice, Shazan apple juice and country apple juice. And Shazan apple juice comes this color and country apple juice comes this color. So that means this one has more reducing sugar in it and this one has less reducing sugar in it. Now this is another diagrammatic representation. Blue solution means no reducing sugar. No, don't say no sugar because this is we're only checking reducing sugar and then it should come to your mind that there's also a non-reducing sugar. So if you just said sugar, that would be incorrect because there are 2 types of sugars, reducing sugars and non-reducing sugars. Green solution means a trace. A green precipitate means a little more. Then yellow precipitate means a little more. And then orange 3 plus and then red brick precipitate. This is a precipitate. Precipitate means that you can actually see there's some little particles in it and that's called a precipitate. So if you can measure the mass of that precipitate, that will also tell you how much is the reducing sugar because more the precipitate, more the reducing sugar. If one has 1 gram or one has 2 grams, so if 2 grams would be more precipitate. Another diagram showing you the Benedict's test for reducing sugar, and this gives you all the colors and increasing concentration of reducing sugar. So the more it is brick red, the more reducing sugar there is. So blue, green, yellow, orange, and brick red. Now coming back to the question, some students were given a 1.0% solution of glucose and some distilled water. They diluted the glucose. They diluted the glucose to produce 5 solutions of different concentration. Dilution means they reduce the concentration. Dilution, I hope you all know that. The students tested each of the solutions they had prepared with Benedict's solution. The concentration of the solution and the results of their tests are shown in table 1.1. Now, B1. The students also decided to test the distilled water with Benedict's solution. Why would they test the distilled water with Benedict's solution? Write the results of this test in table 1.1. So that answer would be blue. Why would it be blue? Because there is no reducing sugar in water. It's distilled water. So distilled water will not have anything in it. There will be no reducing sugar in it. In fact, no sugar in it. Then it says explain why the students tested the distilled water. So what you have to understand is that when they're asking you an explain, then you have to give me, there's only 1 mark. There's only 1 mark for this. There was only 1 mark for this, so you got a mark if you wrote blue here. Then explain why the student tested. Well, basically it's a control, but the control is not going to get you any mark. To show the color when no reducing sugar is present. To show color when no reducing sugar is present, because you have to have a baseline that if there is no sugar, then what is the color of Benedict? Benedict remains blue. It is a blue color. to show color when no glucose present. No glucose present, or if you had said no reducing sugar present. So you got a mark for that. Why the student tested the distilled water? So distilled water is we want to have a baseline that okay, if there's no glucose, this is the color. If there is glucose, then it's going to be some other color. It's not going to be blue, it's going to be some other color. So this is why we tested the distilled water. Now coming to the C part of the question, the students compared the color of their solutions after the same length of time. Like they did it with the fruit juice, then they did it with the other concentrations. Now it says state three other variables that the students should have controlled to make their results comparable. Now I always say when you're doing this question, think you're actually doing this in the lab. So you've got one student takes Benedict solution and takes the fruit juice. Another student decides to take Benedict solution and then fruit juice. Now you see how I have made you understand what was important. What was important was the volume of the solutions. So the volume of the glucose solution is important and the volume of the Benedict solution is important. And then of course the temperature of the water bath is the important thing. So 3 marks. You have to think you are actually doing this in the lab. So how would you do this in the lab? And I just showed you this how you would make a mistake. If we were doing this in the lab and I said, okay, just do the test. Well, every student would take a different volume, but if nobody had told them that you take the same volume, that all the fruit juices should be the same volume, all the solutions that you took, this was no fruit juice. But even when you're comparing it with the fruit juice, you would have the same volume. So if you decide to take 1 ml, then all would be 1 ml, 1 ml solutions and 1 ml Benedict solution. That is a rule that we follow, the equal volume. So this is how I would word it, is I'd say volume of glucose solution should have been kept constant. You can even write that volume. You could have said 2 ml. And volume of Benedict solution also 2 ml. And then the temperature should have been kept constant for all the different solutions that we were testing. So I would have said anything between 80 and 100 degrees Celsius was a correct answer. Then coming to the next part, the students were then provided with a glucose solution X, unknown, of unknown concentration. They repeated the Benedict's test in exactly the same way. They decided that the result was a yellowish orange color. Using the results in table 1.1, suggest what you can conclude about the percentage concentration of glucose in solution X. Explain how you reached this conclusion. Now, how would you reach the conclusion that what is the concentration of something which was what was the color? The color was yellowish orange. So let's look at the table now. So anything yellowish orange would be between 0.3 and 0.4. So the conclusion is it would be between 0.3% and 0.4%. It would be between 0.3% and 0.4%. Why? What is the explanation? The color is intermediate. It's neither yellow, it's not orange, it's yellowish orange. So it is an intermediate between 0.3 and 0.4. Because 0.3 is yellow and 0.4 is orange. So it's something between 0.3 and 0.4. Okay, so this is how we would word it, that conclusion between 0.3 and 0.4 and explanation color is intermediate between 0.3 and 0.4. Okay, then coming to D part 2, the students found it difficult to decide the exact color of solution X after testing it with Benedict solution. Suggest a reason for this difficulty and a method which could be used to improve their confidence in deciding the color. Now the difficulty was that what is the difficulty to describe the color? Because the color is yellowish orange, this would be a difficulty. An improvement is you could use a color chart. If we had a color chart, then we could compare it with a color chart. So difficulty is how do you describe? Is it yellow? Is it orange? Is it yellowish orange? Is it orangish yellow? What is it? So it's a little vague. One cannot be really very sure about what you're talking. And the improvement is, of course, a color chart or one person looking at all the colors, or you have a bigger color palette so that you can understand what that is. So just wording it, difficulty to describe the color because everybody would describe it differently by different individuals. It's a very subjective thing. And the improvement is that you use a color chart. Okay. Now coming to the next part of the question, part E of the question, let's look at that. Part E says describe how the students could use a 1.0% glucose solution to produce 5 centimeter cube of a 0.5% glucose solution. So how would you dilute it? And this is a very new thing in the new this thing. So you have a 2.5 centimeter of glucose solution, and you add the same amount of water in it because you have a 1% glucose solution. So you take 2.5 centimeter cube of the glucose solution of this 1% solution, and then you add what? You add 2.5 centimeter cube of distilled water. Now I give you a very simple example. You have 1 glass of water with 10 grams of glucose in it, and now we add another 9 glasses of water, and the same glucose now is in 10 glasses of water. So this is how you've got to do a simple dilution. You must understand that you can Google this, you can see a lot of videos on YouTube how to do simple dilutions. So 2.5, because you had to make a total of 5 cm cube. That's given in the question. How many did you make in total? So if you have 2.5 centimeter of 1%, now when you add 2.5 water in it, so from 1.0, it has halved. So it has become 0.5. Like if you had a 10% and you took 5 centimeter cube of 10% and then you added 5 centimeter of water in it, so this would become 5%. But you had a 1% solution. So when the 1% solution needs to be halved, so you take the same equal amount. You take 2.5 centimeter of glucose and you add 2.5 centimeter of distilled water. But if I had said you make a total volume of 10 centimeter, then you would have taken 5 centimeter of 10% and added 5 cm of water, and this would have been from 10%, it would have become 5%. So you need to Google this and you need to look up on YouTube simple dilutions, and you would probably understand it if you're not configuring it from my explanation. Now coming to the last part of the question, part F, after the students had tested the glucose solution with the Benedict solution, they noticed that a solid had collected at the bottom of the test tube. They decided that finding the mass of any solid formed was another way of measuring the concentration of glucose in the solution. Suggest how the students could separate any solid from a solution and obtain its mass. Now, if you put some sand in water, it's not going to dissolve. That's going to settle at the bottom of it. So what we needed to do was we needed to filter it. So you would use a filter paper, you would use a funnel, and then you'd put a filter paper in it, and you would pour the solution in it. And if there was any solid, that would stick to the filter paper. So you'd filter it, and then you would measure the mass. So filter it and measure the mass of the solid, or the mass of the solid dried, or the mass of the solid which was present on the filter paper. So all you did was you just poured that solution into the filter paper in a beaker. The solution would all come out, the liquid would all come out, and the solids would stay here on top of the filter paper. And then you would just scrape it off and measure the mass of it on a digital weighing balance. They're very accurate, and they would sort of, handle very small, because this would be a very small, tiny mass. They had finished this question, and this was for 16 marks, and you had to do this question, the first question. This is, of course, a new one in the specimen, slightly different in the previous papers that we used to have. Now coming to question number 2. Carrots are a food containing vitamin C. They can be frozen to be preserved and stored. Some students measured the vitamin C content of fresh and frozen carrots. They then boiled the carrots in water and measured the vitamin C content again. The results are shown in table 2.1. Now, carrots fresh, fresh boiled, frozen, frozen boiled, 5.9, 3.6, 2.5, 2.3, vitamin C in milligram per 100 grams. Construct a bar chart of the data in table 2.1 on the grid. So how do you do a bar chart? Now the rule is, please remember, the rule is always this column, the first column, goes on the x-axis. I've told this time and time again. And this will always go on the y-axis. So you better first label the axis, because this is what you miss out, and that's 1 mark if you miss out. Just writing this vitamin C and writing all of it, milligram, put a slash, milligram per 100 gram, right? So if you've written that, you got a mark for that. Then you have to label the x-axis. Now in the x-axis, what had you to write? You had to write carrots. So x is fully labeled carrots. And then of course you're going to do the bar chart on it. Now please remember in a bar chart that you have to have spaces in a bar chart. So when you look at the 4 marks that were available here, the 4 marks say there's axis fully labeled. So you wrote vitamin C milligram per 100 gram, you wrote carrots here, you got a mark for that. Then linear scale for vitamin C, contain at least half of the grid. So you have to use at least half of the grid, and linear scale for vitamin C, half of the grid in both directions. Then 4 data values plotted correctly. And then all bars ruled and of equal width, and bars not touching. You see, you've got to understand is when you have the touching bars, that's a histogram. That's a histogram in which the bars are touching, and you do them like this. This is a histogram. This is not a bar chart. They had said use a bar chart. So histogram is the one which is something like this. So you are not doing a histogram, you're doing so you must know this difference between a histogram and a bar chart. So you would do this, you can also start it from 0, of course, then you would do it in a different manner, but I have taken the scale from 2 because all the values were 2 and up till 5.9. So I have taken those, but you could have taken it from 0 as well, that would have been correct as well. Then coming to the question of B part 1, state which boiled carrots contain the most vitamin C. Naturally, it would be the fresh one, the fresh one which contained, the fresh one contained about, if you look at it, 5.9. So fresh state which contain the most vitamin C would be the fresh one, which would be the boiled one was also allowed because it says state which contain the most vitamin C. So fresh or fresh boiled would have more vitamin C. So just 2 conclusions the students could reach from these results. Now, what are the conclusions that you get from these results? Boiling decreases the vitamin C, and freezing decreases the vitamin C. So boiling decreases the vitamin C and freezing decreases the vitamin C. That you can see is how in the boiled one, the vitamin C decreased, and in the frozen boiled one, again, the vitamin C decreased. So boiling decreases vitamin C and freezing decreases vitamin C. That is how you would word it. There's not any other word that you could have used. Then coming to the part C of the question, carrots can be cooked by heating them in an oven or by boiling them in water. You want to investigate the effect of these 2 cooking methods, heating them in an oven or boiling them in water. So you can investigate the effect of these 2 cooking methods on the vitamin C content of the cooked carrots. Describe how you would do this investigation. There is a simple test that can be used to measure the vitamin C content. You do not need to know this test. Refer to the vitamin C test in your answer. So this is a rather a long question, and then it's a difficult question because there's 6 marks for it, and the 6 marks you need to get good marks in this to be able to get a grade which is sufficiently okay for you. So 6 marks, the carrots of the same age or the same type or the same species got you 1 mark. Carrot of the same age or same species or the same carrot got you a mark, then the same mass. So whenever you're taking a solid, you always talk of the mass. When you're taking of liquid, you talk of the volume. So same mass of the carrots used, then both cooking methods should be used. You must use it. You must do the oven method, and you must use the boiling method. So both cooking methods, oven and the boiling method, should be used, and the same temperature for the same time. So you cooked it for the same temperature and for the same time. Then the vitamin C test was used on the content determined before cooking, and then the vitamin C test was used on the content determined after cooking. So before you tested it, before you did any of the procedures, the cooking procedures, you tested it before that, and then you tested it after that. And then the last point which you can write in any one of these questions is you repeat the procedure to see if similar results are obtained. You see, if you repeat it, you did it once, and then the second time you did it and something else was the answer. So you need to do it a third time to see which one was the correct one. Was it the first one the correct one or the second one? So you always repeat the experiment at least 3 times so that then you can calculate an average and then you can come to a good enough conclusion. So this is what you have to do for these sort of mark schemes, and these are the sort of questions which sometimes you find difficult, but some of you are very good at it. Now you can see the written mark scheme as well. I don't write the mark scheme initially because some of you have commented in my videos that please miss, let us do the question first and then we want to see that. So I give you time, plus you must take this paper out and from the internet and do the paper before you start watching this video. So carrots of the same age, same mass of carrots, both cooking methods used. Now this was given in the question, but you'll get a mark for that. Same temperature, same time, vitamin C test before cooking, vitamin C test after cooking, and repeat 3 times to see if the same results obtained. So repeat procedure for 3 times so that you can see if it's the same result and there are no anomalous readings. There could be an anomalous reading and three readings show something else and the fourth reading shows something else. So then you know that what was the trend. If the 3 results show this, so it's better even if you say 5 times. If you say 5 times in A levels, we always say 5 times because that gives you a much better measure, because one could be just a mistake or an error or a human error or a experimental error, and you could be getting a different result. So even if you do it 5 times, that is a very good way, because if one was wrong, then you know what is the general trend. So this was the mark scheme for the second question, and I hope everybody is clear. If you have any comments, please do let me know. Now coming to question number 3, the potato plant is a plant that stores starch grains in the cell. Show some of these starch grains as seen under the microscope. Identify the structure D. Now the structure D, as you can see, if it's a starch grain, then it has to be potato plant, it has to be the cell wall. Draw the starch grains labeled E, F, and G as they appear in figure 3.1. Grain F should be at least 60 millimeters long. Most of you always say that, Miss, we don't know how to draw and drawing is very tough. It's only 3 marks for the drawing, and you can even get this wrong. You can at least get 2 out of 3. So basically, why are you getting the marks? Is clear continuous lines, no shading. And grain F at least 60 millimeter long, because that's given in the question, and the grains should be touching each other. Correct relative proportion of all these 3 grains. I mean, they must look the same. Like for instance, if you look at this diagram, E, F, and G. So what is E? E is this. And then this is a bit bigger than this, but it's not that big. And then G is this. So it must be the same proportions. You can't just draw one little tiny little thing and then this huge thing and then this tiny little thing. Well, this is not proper proportions. Just look at both of them. So they must be in the same proportions. That's what they're saying, correct relative proportions of all these grains. On your drawing, draw a line to indicate the maximum length of grain F. Measure this length and record it. That's 69 millimeter. You can see here I've drawn that line. So it's 69 millimeter. The actual length of F is 0.03 millimeter. Calculate the magnification space for working. So you can see the working of this is 69 divided by 0.03, and this gets a magnification of 2300. Now describe how to prepare a slide of potato tissue to observe starch grains as clearly as possible under a microscope. Scraping from the cut surface, drop of iodine solution to stain, add cover slip, remove the air bubbles and excess stain removed. So basically how a piece of potato has to be observed, this is of course a part of the syllabus that you had to examine how to prepare a slide for the microscope. So scraping from the cut surface, you add some dye because you know cells don't have any color, and in order to stain it, and then you add the cover slip, and then you remove the air bubbles, remove the excess stain, and you examine under the microscope. Now that completes this specimen paper, and I hope this is helpful to you all, and you all can now I will of course be doing the more of these papers online for you all, so that you can watch the videos and understand how you're going to be doing the paper, our alternative to practical paper. So best of luck and all the best, and thank you very much.